Hello everyone, welcome back to the channel The Biology Club. So today we are going to see the second part of the animal kingdom of class 11th, myself Jyoti Vishwas. So in the last video we have seen about uh, till the organ system level of organization. Now there are more things which we are gonna learn in this particular video. So let's start with that. So let me introduce to you the diploblastic organization. Okay, the diploblastic organization. Now, from the name itself, you can see diploblastic. D. So something is there where two things are there, right? So you have to understand that. So here two things are present. Now let me tell you what. So in animals, cells are arranged differently. Okay, so the first arrangement which we are going to learn about is about the diploblastic organization or diploblastic arrangement, right? So in those animals, in those animals where the cells are arranged in two embryonic layers, in two embryonic layers. So when the animal is in embryo form, right? When the animal is in embryonic stage, so at that time, the cells in it are arranged like this. We are talking about that during the animal, when the animal is in embryonic stage, how the cells are arranged. Okay, so here the two embryonic layers will form. First one, the external one means the outermost one. The external layer, let me write it down, layer also, is known as ectoderm. Okay, the external layer is known as ectoderm and the internal layer is known as endoderm. Fine. So let's imagine it first, how it will look like, right? Just give me a second. Let me change the color. Yeah. So let's draw the outer layer with purple color. Okay, so this is the outer layer of the cell, which is the ectoderm, right? Then you have innermost layer. You have the innermost layer. Let's draw it with some other color, which is like this. This is the endoderm, something like this. Now, there is one little twist. In between these two layers, there is one undifferentiated layer. So the scientists, they have not yet figured out how the layer is situated there, what is the shape of that particular layer between these two things, and they have named this particular layer as there is one layer like this, which is undifferentiated. Cells are not visible correctly, so they call it mesoglia. Mesoglia. Okay, and let me show you how it is there in your book. You might have seen it already. Something like this. Okay, let me rub my figure. It is overlapping. Right? Yeah, this is how it is look like. You can see here, outermost layer, the purple color layer is the ectoderm. Then the inner grayish color layers are the endoderm. And in between these two layers, we have one mesoglia also, right, which is undifferentiated layer present between ectoderm and endoderm. Fine. Okay. Now let's talk about triploblastic organization. Triploblastic, now three things are here. Okay, triploblastic means three things are here. Now here the animal in which the developing embryo has a third germinal layer between the ectoderm and endoderm. So first external layer is known as ectoderm. We all know that. Then the inside one, the internal one, is known as endoderm. Here, instead of mesoglia, these cells are very prominent and uh, visible nicely. Okay, so that layer is known as mesoderm. This is situated between ectoderm and endoderm. Okay, now this type of animals are known as triploblastic animals. Examples from platyhelminthes to chordates, everything we know comes under triploblastic organization. Fine, let's look how this organization is look like. This is how it will look like. Okay, outermost is the endoderm, outermost is the ectoderm, innermost is the endoderm, and as you can see, in between these two layers, we have one more layer. That is the mesoderm. So how prominent it is, right? How beautifully we can see it. So that's what. Fine. Okay, so let's start with the coelom now. Okay, so what are coelom? So, the body cavity which is lined by mesoderm is known as coelom. Basically, these are the body cavities lined by mesoderm. So the mesoderm we have seen in between the layers, right? Ectoderm and endoderm. So this particular body cavity is done by mesoderm. Okay. Now let me show you the picture here so that the things will be clear to you. Okay. So the first one, as you can see here, first one which is known as a coelom here, the um, coelom is absent, means the body cavity is absent. Okay, so this type of organism is known as organization of cell is known as a coelom, which is found in Polyphylla cylindrita, platyhelminthes. Okay, and the animal or the organism is known as a coelomates. A coelomates. Now there is next one, which is pseudocoelomate. Okay, so now in some animals, the body cavity is not lined by mesoderm. Instead, mesoderm is present like a pouch. You can see here, these mesoderms are present like pouches. Okay, now this type of organization leads to pseudocoelomates. Okay, fine. So the animal is known as pseudocoelomates and this organization is known as pseudocoelom. Fine. Okay, now see this thing. This particular thing. Here, as you can see, this is the true coelom, the perfect coelom we are talking about, the cavity. So this is the cavity and it is lined by mesoderm. Here. Okay, the yellow one is mesoderm. So this particular thing is lined by mesoderm. So this is the perfect coelom we are talking about. This is the perfect, body cavity. Now, animal which process coelom is called coelomate. For example, from annelids to chordata, each and every animal consists this type of organization. Fine. Okay. Let's move on to the next topic. Segmentation. Okay, segmentation. So, what is segmentation? See, segments. Segments. When the body is externally and internally divided into segments with serial repetition of at least some organs, we call it segmentation. Fine. So like this. For example, earthworm. Have you seen earthworm? Of course, you have seen earthworm, right? So this is the organization now. It is dividing this particular portion is completely dividing externally and internally. That particular segment has been made there. Fine. Yes. Now, example, I have given you the example of earthworm. So, in earthworm, the body shows this pattern called metameric segmentation and the phenomena is known as metamerism. Okay, so this is how the body of earthworm will look like. Okay, So this is one organ has been divided here. Again, the next organ is divided like that. Again, the next organ is divided like that. Okay, so it is justifying the definition. The body is externally and internally divided into segments with serial repetition of at least some organ, not all, some organ we are talking about. Okay, so this is the cavity we have just learned now, which is the coelom, right? Now, in arthropods also we can see metamerism, but only external. Let me write it down. In arthropods, metamerism is external only. External. And in vertebrates, it is internal. That's it. In vertebrates, it is internal. Fine. Okay, let's move to the next slide. Now, we will talk about a very important part, that is notochord. Okay, now animals with notochord are called chordates. Those without it are known as non-chordates. So let me write it down for you. Animals with notochord are called chordates. And without notochord, animals without notochord are called non-chordates. Okay, for example, Porifera to echinoderms, everything comes in between are what? Non-chordates, right? So, so let's see how it is look like. Let me rub it for you. How it is basically look like. Notochord, it is situated dorsoventrally. Let me show you how. Okay, so here it is mesodermally derived rod-like structure formed on the dorsal side during the embryonic development in some animals, not all, in some, right? Like this. So as you can see, this layer is the notochord, and above that there is one nerve cord. Now, eventually, in human beings, if we are talking about, so during the time period when the embryo will become the fetus, this notochord, in above that you can see the nerve cord, right? So this nerve cord will become the spinal cord slowly. Fine. Now let's see what we have learned till now. So we were learning about this particular thing, the kingdom Animalia, which is the animal kingdom, right? Now we have seen the different level of organization: cellular level of organization, tissue organ system level of organization. So always remember that the cellular level of organization will have a symmetrical symmetry, and the body cavity will be, obviously it will be absent, right? A coelomate will be there. There are coelomate. Example, porifera. Tissue organ system, tissue organ or organ system, we have two types, that is radial symmetry and bilateral symmetry. In radial symmetry, you have a coelomate, that is coelenterata, ctenophora, right? In bilateral, you can see there can be a coelomate, pseudocoelomate or coelomate. Now, if we are talking about a coelomate, that is platyhelminthes, pseudocoelomate, that will be Escalmenthes, then coelomate, Annelida, arthropoda, mollusks, echinoderms, hemichordata, chordata. Okay, this all comes under this. This is what we have learned till now. If you will memorize this particular table, you will get it. Now, in examination, they will never ask you define radial symmetry, define bilateral symmetry. They will give you one organism or one phylum and they will ask you to guess it, what type of symmetry it will have. Okay, so you have to learn each and every phylum very seriously now. So these were the basic introduction from my side. We will start the next thing in the next topics or the phylum we are going to learn about. We will see in the next coming video. Thank you. Have a nice day.